Hi everyone, my name is Ain Ashkimita Asmawi, and today I would like to briefly introduce the objective and overview of our field-based environmental biological learning session. So, this hands-on program is designed to enhance student understanding of environmental biology and ecological system, emphasize learning through direct field experience rather than just a classroom theory. So, this is our field visit schedule and also what we got from this field trip. Before we begin with our collecting data, let's look at objective. First, to calculate and describe the following descriptive statistic, which is mean, variance, and also standard deviation. It also to understand the concept of correlation, and lastly to determine the relationship between two variables. So, in this practical, we only use three material, which is leaves, ruler, and also graph paper. Set 1, mean for leaf length is 8.33, variance 0.2254, and standard deviation 0.4748. For the leaf width, mean 5.57, variance 0.6334, and standard deviation 0.7959. Set 2, mean of leaf length is 20.3, variance 12.8622, standard deviation 3.5863. For the leaf width, mean 5.73, variation 0.4378, standard deviation 0.6617. For the last set, mean for leaf length 20.39, variation 2.1423, standard deviation 1.46. Leaf width, mean 5.05, variance 0.2138, standard deviation 0.46. Answer for first question: if the mean leaf length varies across site, likely due to species differences or environmental factors like light and water. Some sites share similar length, indicating uniform condition. In contrast, leaf width remain mostly consistent, suggesting stable growth factor with slight difference, possibly due to species trait or environmental adaptation. Second answer is variance measure how much value in data set differ from the mean. When we say the leaf have different measurement, it means their size vary. This is what variance capture. A high variance show wide differences, while a low variance means the size are more uniform. Standard deviation are the square root of variance, show the average amount of deviation in the same unit as a data. So, for the last question the answer is collecting multiple set of leaf width and length measurement help capture natural variation within and between species and show how environmental factors like sunlight, water, and soil affect growth. It allows researchers to identify trends, compare average measurement variability, and make more accurate generalizable conclusion about leaf tree.
Assalamualaikum and hi, my name is Nur Azin Shafrina binti Muhammad Suhaimi and my student ID is 2025597939. I'll be explaining about practical 2, which is the factors that affect the distribution and abundance of organism. Let's start with the objective. For the first objective is to introduce the simple concept of quadrat. The second one is to enhance the power of observation. The third one is to record the difference in plant distribution within the quadrats, and the last one is to infer from natural observation the possible factor that affect the distribution and abundance of organism. Let's take a look at the result. For the first quadrat, we found 5 species. The first one is Shorea acuminata. The total number of individuals is 1 and the density is 0.04. Next is Pinanga acaulis. The total number of individuals is 3 and the density is 0.12. The third one is Cyanometra malasensi. The total number of individuals is 1 and the density is 0.04. Next is Liquala orbiculari. The total number of individuals is 1 and the density is 0.04. The last one is Garcinia arthroridis and the total number of individuals is 1 and the density is 0.04. For the second quadrat, we also found 5 species. The first one is Trodesanta zebrina. The total number of individuals is 7 and the density is 0.28. Next is Araceci Johann Tegesmania peracensis. The total number of individuals is 3 and the density is 0.12. The third one is Dry Balanops oblongifolia. The total number of individuals is 1 and the density is 0.04. Next is Shoria microphylla. The total number of individuals is 1 and the density is 0.04. And the last one is Pentaspadon motley. And the total number of individuals is 1 and the density is 0.04. Okay, next let's go to the post-lab question. For the first one is define the term quadrat in practice. Quadrat is a small, typically rectangular or square area used to sample vegetation or other organism in ecological studies. In practice, quadrat are employed in various sampling techniques such as quadrat sampling or plot sampling to obtain quantitative data about a species composition, abundance and distribution within a defined area to analyze and compare different ecosystems. For the second question, what are the two basic requirements of the quadrat technique? The two basic requirements of all the quadrat technique are the area of the quadrat counted and the number of organisms of species in a quadrat. Next, let's go to the question number 4. Problem with spatial scale often make it difficult to directly estimate average densities in habitats such as large forested areas or in aquatic habitats where one cannot directly see the animal or plants of interest. Explain the issues concern ecologists in conducting in sampling method. Okay, for the answer is, since it is usually impossible to sample an entire population within a study area, one must take a smaller sample of the community or a population of interest. The sampling method needs to have the details about the densities and dispersion patterns of the organism or types of the species in the population. Then, the size of the sampling plots, where the sampling plot will be placed, and the number of the sampling plots needed to be conducted.
Putrajaya wetland use 24 engineered cells with over 70 native plant species to filter water naturally. These plants help remove about 82% of nitrogen and 84% of phosphate before the water flow into the lake. So this is the function of wetland plants, which is immerged plant that trap sediment and absorb nutrients, slow water flow for better filtration, support beneficial microbe and also prevent soil. For floating plants, it absorbs nutrients from the surface, limit algal growth by shading water, boost oxygen and remove heavy water pollutant. So here's our collecting natural water filter plant. First, its botanic name is Phragmites carca, we known as a rumput gedabong, and second is Eraucaulon longifolium, its botanic name, and known as a rumput butang. Third is its botanic name is Lepironia articulata, and we know as a purun.
Okay, next, the botanic name for this species is Hangwana malayana. Okay, as for the conclusion, our trip to Taman Wetland and Taman Botani Putrajaya was a great hands-on experience that helped us better understand how ecosystem and biodiversity work. By identifying different species and doing practical activities, we learned how things like environmental factors and natural variation affect where organisms live and how many there are. The trip didn't just teach us more about science, it also showed why it is important to take care of the environment and manage ecosystem wisely. Overall, it was a fun and meaningful learning experience that helped us connect what we learned in class to the real world.